Hey guys, in this video we'll be talking about the urea cycle. This will be the last video I make because we didn't go over amino acid metabolism or lipids. I might do one for techniques, but there's not a PowerPoint out there for me to follow from, so I might just answer questions on that, but I probably won't be able to make a video on that. So this is probably the last one. So we'll go over the urea cycle. Basically, what the urea cycle is when you have an amino acid. Lower this a bit. When we have an amino acid, we have this amino group, and we also have a carbon skeleton. When we want to use the carbon skeleton in glycolysis or in TCA, we don't want this amino group with it. We just want to be able to use the carbon skeleton and get the energy out of that. So what we do is we run this transaminase reaction. It's coupled with alpha ketoglutarate and glutamate. So this is similar to TCA stuff. NH3 is then given to alpha ketoglutarate, so this becomes glutamate, and then the carbon skeleton becomes an alpha keto acid. So this can be run with different amino acids. Usually the ones you'll see are alanine, and then pyruvate would be your alpha keto acid, or you can also see it another way is aspartate and alanine are amino acids. This is non-hydrophobic, and this is one of the acidic amino acids. Aspartate can then be converted to oxaloacetate. So those are the two you'll see, but they can also be run with different amino acids. Once we make glutamate, so glutamate, remember, has the amino group. We run through a reaction called glutamate dehydrogenase. NH3 is sent to the mitochondria. We regenerate alpha ketoglutarate to be able to run the transaminase reaction again. This is coupled with NAD being reduced to NADH. Glutamate is oxidized to alpha ketoglutarate. This can also be run with NADP as well. But the whole point of this is we can generate NH3, which then goes through the mitochondria. We do a couple more reactions, and then eventually this NH3 will ends up in urea, and then it's excreted. Okay, so if you'll see, the organs that will do these it's gonna mainly be the liver, but then some other tissues express the enzymes, but they don't run the urea cycle. So the liver will be where most of the urea cycle occurs. You'll see that this happens in the mitochondria and the cytosol. So we start in the cytosol, we send NH3 in the mitochondria, and then eventually we do the carbonyl phosphate synthetase. We make citrulline over here. Citrulline is exported out, and then it goes through the cycle, and then when we're done, we make ornithine, which then exported into the mitochondria, and it continues the cycle. Runs very similarly to TCA in the way that it works, but we have these transporters, which we'll talk about, that help us transfer substrates from the mitochondria to the cytosol. Okay, so that's kind of just a general overview of what the urea cycle is. Okay, so nitrogen uptake. So remember, in plants, bacteria, bacteria, they take N2, NO3, NO2, and NO3, sorry, and then they nitrogen fixate, send it to plants, and then plants are able to put those in forms that us mammals and other mammals out there can use. And then, so it's eventually fixed to NH3. Eventually, this will end up in amino acids, which we get from our diet. So the amino acid, the amino group, goes into the urea cycle, and it's excreted, and then the carbon skeleton can then be used to make energy for the cell. So you have glucogenic amino acids and ketogenic amino acids. Glucogenic will end up here, then ketogenic end up as ketone bodies, but they're both used as the carbon skeletons for both is used to make energy for the cell. So, like I said, amino acid breakdown, carbon skeleton is oxidized, and then we get rid of the nitrogen through the urea cycle. Okay. So let's go over the transaminase reaction and why we need to get rid of ammonia. So transaminase reaction, like I briefly mentioned, you have an amino acid, you have alpha ketoglutarate. So this amino acid can be any of the amino acids, but the ones you'll see, like I said, are alanine and aspartate. You make an alpha keto acid, So if you use alanine, you make pyruvate. If you use aspartate, you'll make oxaloacetate. Transaminase, we put the NH3 on alpha-ketoglutarate, we make glutamate. And then we regenerate alpha-ketoglutarate to run the transaminase reaction again. And in that process, we reduce NAD to NADH, and then ammonia gets sent to the mitochondria. Okay? So the reason why we can't just excrete it as ammonia, because it's toxic. So the nitrogen cannot stay in ammonia form, otherwise it could cause a lot of problems in the cell. So we convert it into urea, which is easier to excrete, and it's not toxic. You'll see that urea also has two nitrogen groups. We'll talk about where the other nitrogen comes from. So let's talk about why we cannot just excrete it as ammonia. So ammonia exists in your body at concentrations less than 50 micromolars. If it starts to accumulate, it can act on the nervous system, and this can cause drowsiness, seizures, and coma. Eventually, you can have complications such as VHE, and this can lead to unusual complications in the nervous system, and then this can cause decreased level of conscientiousness. Okay, so let's talk about the pathology of how it could cause this. So remember, so this is the nervous system, but it can also cause pathology in other tissues. So this is your neuron, this is astrocyte. Remember, they exchange substrates. So one thing that they exchange between each other is, remember, we talked about lactate. The other thing we talked about is they can exchange glutamic acid and glutamine. So glutamic acid and glutamine are two amino acids. Glutamine is just glutamic acid, but it has an extra amino group. Okay. So because when we convert from glutamic acid to glutamine, we use NH3. So if NH3 starts accumulating in the nervous system, this will increase the levels of glutamine, and then it will decrease glutamic acid because more glutamic acid is being converted into glutamine. This will affect the concentrations of both glutamic acid and glutamine, and then this can affect other parts of the cell. Because remember, glutamic acid or glutamate, and so I didn't mention this, but glutamate just has the COO minus in carboxylic acid group, and then glutamic acid is the same thing. It just has the COOH, so it's the protonated form, and glutamate is the deprotonated form. Okay, so this GLU refers to the same amino acid, so it can be glutamate or glutamic acid depending on the pH of whether it's not protonated or not. So remember, glutamate can be converted into alpha-ketoglutarate, and this can enter into TCA. So if you change the levels of glutamate, this will change levels of alpha-ketoglutarate. It'll affect TCA, and that's how you get all your complications in the nervous system. Okay, so after we run through the transaminase reaction, we run through the glutamate dehydrogenase reaction. We send ammonia into the mitochondria, and then ammonia joins in with bicarbonate. We use some ATP, and then we run through carbamyl phosphatase synthase, which then makes carbamyl phosphate, and then carbamyl phosphate starts the urea cycle. So we start with carbamyl phosphate down here and ornithine. So we take carbamyl phosphate, ornithine, and then we make citrulline, and that's how we start the urea cycle. And our goal is to eliminate the nitrogen that was originally in the amino acid. And we'll talk about how the urea cycle is also connected to other things in the cell, like TCA, for example. Okay, so let's talk about glutamate dehydrogenase first. So glutamate in the process is oxidized to alpha-ketoglutarate. So this reaction is also reversible, so it can run the other way as well. Same thing with the transaminase reaction. It's also reversible. They're both reversible. They can run either way. Another thing I'll mention about the transaminase reaction is this is very similar. This is the same reaction we talked about in the malate-aspartate shuttle. So if you go back to the malate-aspartate shuttle, there is usually a transaminase reaction there, and it's referring to this reaction. Okay, so if you go there and briefly look at that, it will look very familiar. Okay, so back to glutamate dehydrogenase. We take glutamate, convert it, oxidizes to alpha-ketoglutarate, and then in the process, we reduce NAD to NADH. Ammonia is sent into mitochondria. So this reaction can use NAD or NADP. NADP is not really realistic, but theoretically it could use it. But most cells will just end up using NAD and NADH. NADH can then also be sent into ETC to supply more energy. Okay, so now that we got rid of the ammonia, it's in the mitochondria. Actually, I have a slide about glutamate dehydrogenase. So it's a hexameric structure. It can be, so ATP and GTP will promote glutamate formation and inhibit alpha-ketoglutarate formation. Let's see why that is. It doesn't really have much to do with energy. It's mainly because we use ATP, and we'll see another reaction in Urea cycle also uses ATP. So ATP drives them this way, right? If ATP starts accumulating on this side, it will kind of negatively feedback, inhibit the reaction before it. So we'll tell the reaction, hey, we have a lot of ATP, we'll favor the other direction. Because it's one of the substrates used with ammonia. So if ATP starts to accumulate, it will push all of this in this direction, okay? If ADP starts to accumulate, it will push everything in this direction, so we'll favor the glutamate to alpha-ketoglutarate reaction, okay? So ADP accumulates, we go this direction. ATP accumulates, it will favor the other direction. So if ATP accumulates, we'll favor this direction, okay? So that's why ATP or GTP will promote glutamate formation and inhibit alpha-ketoglutarate formation. And then ADP does the opposite. It will promote alpha-ketoglutarate formation because we're driving it in this direction. Okay. If you want to think about it about Le Chatelier's principle, if this increases, it will shift the reaction in this direction, and then this will shift the reaction in this direction. Actually, wait. Sorry about that, Dave. Just forget what I said. That's not that. You shouldn't look at what I said. But if ADP accumulates, we'll favor this direction. Okay. And then NADH can also inhibit alpha-ketoglutarate formation. So if NADH starts to accumulate, it will go back and inhibit this. So we'll inhibit alpha-ketoglutarate formation. Okay. Okay. Next, you can have also different isoforms. So glutamate dehydrogenase 2 is high in the nervous system, and it's not inhibited by GTP. So because it's not inhibited by GTP, this allows response to a lot of glutamate levels. Okay? So if you have a lot of glutamate, you'll make a lot of ATP. ATP will then go back and inhibit this reaction. So glutamate dehydrogenase 2 allows us to have more glutamate, and then it doesn't allow for this inhibition to occur. Okay, so back to the ATP. Right, because, okay, so I'll explain it better. If ATP accumulates, it will go back and inhibit this reaction, and this will favor glutamate, okay? And then ADP would do the opposite. Because you have ADP accumulating, that means you don't have ATP to inhibit this. So glutamate dehydrogenase is running in this direction, favoring alpha-ketoglutarate. Sorry if I confused anyone when I was trying to explain this. Okay, so ATP just goes back to inhibit it. So if it's inhibited, if there's a lot of ATP, it will be inhibited, glutamate will be formed, and then ADP would do the opposite. Okay, so urea cycle overview. So we have the free ammonium. It's condensed into bicarbonate. So that's the reaction we'll talk about in a sec. Eventually this is converted to urea, and then it's excreted by the kidney. Okay, so this is the carbamyl phosphate synthetase. So this is the next reaction, which is down here. So we take bicarbonate, we take ammonia, we take ATP. So this reaction happens in the mitochondria. And then we make carbonyl phosphate, which is able to start the urea cycle. This is the rate limiting step. Like I said, we use NH3, ATP, bicarbonate, and this makes carbonyl phosphate and ADP. And it has 3 parts. Okay. So let's talk about the mechanism of it. So this is bicarbonate down here. This is ATP. So bicarbonate will start by attacking ATP. We get a carboxyphosphate intermediate. So this is very similar to pyruvate carboxylase, if you remember. Carboxyphosphate is then attacked by ammonium. A phosphate group is a pyrophosphate group is no just one. Yeah, one phosphate is eliminated. We make carbamate, and then ATP is attached to carbamate, and then we make the carbonyl phosphate product. Okay, so the carbonyl phosphate, we have this NH3 that we added, this CO2 group that we started from bicarbonate. So this came from bicarbonate. And then we also have this phosphate that came from ATP. And then we're left with ADP. Okay. Another thing I'll mention is this phosphate is linked like this. So this is an anhydride. And we'll see why this is important in the next slides. Okay. So now that we make carbonyl phosphate, let's talk about the carbonyl phosphate synthesis enzyme first. So there's 2 isoforms. CPS1, so this is the one that we're talking about in this lecture. It's used in the urea cycle in the mitochondria. Other ones are used in pyrimidine biosynthesis, and it's in the cytosol. So pyrimidine, these are like your C and T base pairs in nucleotides. So CPS is used in these. Okay, so we're not going to talk about that. We're talking about CPS1 in this lecture, but it's important to be aware of CPS2 as well. Okay, so after that we have ornithine transcarbamylase. So we'll take the carbonyl phosphate we made, we'll take ornithine, and then we'll make citrulline, and we'll be able to start. So like I said, it takes ornithine, carbonyl group from the carbonyl phosphate, and we'll make citrulline. So this is the carbonyl phosphate. Like I said, this O is linked to phosphate, and this is the structure of the phosphate group. So this is an anhydride. This anhydride is very high energy, and it's able to drive the reaction. So if you were ever asked about why ornithine transcarbamylase is driven in this direction toward citrulline, towards citrulline, it's because of the high energy intermediate. Okay. So it has, do I have the mechanism for it? No, I have the mechanism for the next one. Basically, the NH2 group from ornithine attacks the anhydride here at this double bond oxygen, and then this acts as a leaving group. The oxygen with the phosphate, you get carbonyl phosphate left. No, you don't get carbonyl phosphate. I'm sorry, this is carbonyl phosphate. You get the phosphate with 4 oxygen attached to it. Ornithine is a non-standard amino acid, and it can also be used to synthesize other things that we'll talk about in the later slide. So after we make citrulline, we get citrulline and aspartate. We use ATP, so this is the 3rd ATP that we used. So you'll see why a lot of ATP will favor glutamate. If there's a lot of ATP, it will favor the opposite direction. So this reaction is done by argininosuccinate synthetase. It takes citrulline, aspartate, and we make argininosuccinate. I think I have the mechanism for this one, yeah. So this requires ATP, of course, I just mentioned. So citrulline attacks ATP, pyrophosphate is eliminated, and then citrulyl AMP is formed. So this is our intermediate, and then aspartate comes in, attacks citrulyl AMP, AMP leaves, and then argininosuccinate is formed. Okay, so this is actually where the second nitrogen comes from. It comes from aspartate. So if we go back, we have 1 nitrogen over here that comes from an amino acid, which is down here, the green one in urea. It's this one. Then we also have this nitrogen that appears out of nowhere because urea has two nitrogens. Like this nitrogen comes from here. So if you're asked about where the second nitrogen comes from, it comes from aspartate in the argininosuccinate synthesis reaction. Okay. So now that we made argininosuccinate, our next reaction is argininosuccinase, or another name for it is argininosuccinate lyase. Basically, this reaction takes argininosuccinate over here, we'll cleave it, and then we'll get fumarate, and then we'll get arginine. Okay, and then arginine is eventually converted to urea. Okay, fumarate. Remember fumarate. So this is an intermediate in TCA. So it can be converted to malate and then oxaloacetate and then enters gluconeogenesis. So this is fumarate. Remember, we can run fumarase, same TCA enzyme, except we're in the cytosol right now. We make malate. Malate can run through malate dehydrogenase, and then we can make oxaloacetate, and then this can go into gluconeogenesis. So if we go back, this is not TCA. They're the same enzymes. They're the same enzymes expressed in the cytosol, but it's not TCA because this is the cytosol. So that's why this oxaloacetate ends up going into gluconeogenesis and not TCA. If you remember, we can run the malate aspartate shuttle. Malate aspartate shuttle is from malate to oxaloacetate to aspartate, and this can then enter the mitochondria and run into TCA. If you want to enter the mitochondria and do TCA, you would have to run through malate aspartate shuttle, but if that's not mentioned, oxaloacetate will end up in gluconeogenesis. Okay, so these are the same enzymes, except these are the cytosolic forms of the ones in TCA. Okay, so back to where we were. So we had argininosuccinate cleaved to fumarate, and then arginine, and then arginine goes into arginase. Then that's how we make urea, and then we make ornithine. Ornithine then enters the mitochondria, and it can continue the cycle. Urea is then sent to the kidney, and then it's excreted from there. Okay, so arginase, we take L-arginine, and it's converted to ornithine and urea. Ornithine continues the cycle. Urea is excreted. This is the structure of ornithine. This is the structure of urea. I'm not sure if you guys are expected to know the structures for the urea cycle. When we took it, it was we were expected to know some of them, and I think Dr. Shriver told us which ones we needed to know, so I would ask about that, but I'm not sure. I don't think you're expected to know all of them. Just I would ask if you're unsure. Okay, let's talk about how the urea cycle and TCA are related to each other. Okay, so if we go here, I know it's confusing, but we'll look at it one part at a time. So this is alpha-ketoglutarate. This is an amino acid, this is alpha-keto acid, and this is glutamate. So remember, this is a transaminase reaction, and this can happen with aspartate, oxaloacetate, or you could run alanine. Those are the most common ones you'll see, to pyruvate. You'll make your alpha-keto acid, so this can go into glycolysis or ketone bodies or whatever, and then you make glutamate, and then glutamate goes back to alpha-ketoglutarate. So this is glutamate dehydrogenase. By the way, the key is down here. If you're unsure which ones are which, they're color coded. This is glutamate dehydrogenase. We convert back to alpha-ketoglutarate, and then ammonia is made, and then carbonyl phosphate synthetase takes ATP, takes bicarbonate, takes ammonia, and then we make carbonyl phosphate. Carbonyl phosphate comes in with ornithine, and then we make citrulline. Citrulline is exported out of the mitochondria into the cytosol. We take citrulline and then aspartate, and then we make argininosuccinase. Then this can be eventually converted to arginine, and then arginine is converted to urea, and then it's cleaved to urea and ornithine. Ornithine continues the cycle. So when argininosuccinase is cleaved, we get arginine, and then we also get fumarate. Fumarate can be converted to malate, which converted to oxaloacetate. So these are the cytosolic versions of the TCA enzymes. And then oxaloacetate can be converted to aspartate. So to convert from oxaloacetate to aspartate, so this is your transaminase reaction. reaction. Oxaloacetate is an alpha keto acid, and then this is your amino acid. So if we go back, it's this reaction just running the other way. So oxaloacetate is converted to aspartate. Aspartate can then continue the cycle. In that process, glutamate is converted to alpha ketoglutarate, and then you can convert back to alpha ketoglutarate using another transaminase up here. So transaminase reactions, don't confuse them because they can run with whatever amino acids you're trying to use. Okay, so malate. So if you're asked about this on any questions, because I missed this last year, what are the four substrates that connect TCA and urea cycle? It's fumarate, malate, oxaloacetate, and alpha ketoglutarate. Those allow the exchanging between urea and TCA. So if you're asked about that, those are the four substrates. Oxaloacetate can be transaminated to aspartate, so we already talked about this. So if we go back to here, so oxaloacetate can actually be converted to aspartate, and in that process, glutamate can be converted to alpha Kg. So this reaction up here can be run down here as well. It's all on the side as well. And then we can supply aspartate to get more of the ammonia out of the cell. Okay? So that's what this means. The oxaloacetate can be transaminated to aspartate and put into the mitochondria. So it can be converted to aspartate, which can go through the urea cycle, or it can be converted to aspartate and then run through the malate aspartate shuttle. So this would be your malate aspartate shuttle when it says put into the mitochondria. Put into the mitochondria, and then it runs through that and then enters the mitochondria to supply NAD for glycolysis to run in the cytosol. And then it also sends substrates to TCA to run in the mitochondria. So there are cytosolic versions of fumarate and malate dehydrogenase. So they're talking about these over here. This is supposed to say fumarase. So this is fumarase, and this is malate dehydrogenase. Okay, then malate can enter the mitochondria and run TCA. So that's the malate aspartate shuttle again. Okay. So let's talk about energy balance. So remember, we get one NADH up here. So that can go into ETC, and then it can be used to make energy. And then when we go from fumarate to malate, then we'll eventually go to oxaloacetate, and then oxaloacetate can be transaminated to aspartate. Remember, from malate to oxaloacetate, we make NADH. So we make second NADH down here. We'll make another one up here. So we make two NADHs, and we use 3 equivalents of ATP. So remember, one NADH gives you 2.5 ATP. So if we have 2 NADH times 2.5 is we technically make 5 ATP, and we used 3 equivalents. So technically we generated energy for the cell when we run urea cycle. But most of the time, you're not going to run urea cycle to make energy. You're just running it to excrete ammonia. But it's kind of nice that it allows the cell to excrete ammonia, and you also get these NADH that then can be converted into energy in the electron transport chain. Okay, so let's talk about another use for the ammonia cycle. So remember, muscles, they use amino acids. Mainly when you talk about what uses amino acids for energy, you'll talk about muscles. So we have this amino acids. We want to use it for energy in the muscle. We convert it to alpha keto acid. So this can then enter either glycolysis, or if it's glucogenic, it will enter glycolysis. If it's ketogenic, glycolysis, and then it can also be used to make ketone bodies. Actually, most the liver won't make ketone bodies, so most of it, the liver is what makes ketone bodies, not the muscle. So most of the alpha keto acids will go into glycolysis, and in that process, the amino group that was on the amino acid is given to glutamate. So we need to get rid of that somehow. But remember, only the liver. So this is your liver. This is muscle. Remember, only your liver does the urea cycle. So glutamate is then converted to alpha-ketoglutarate back, and in that process we convert pyruvate to alanine. Okay, so pyruvate is your alpha-keto acid, then alanine is your amino acid. And this is another transaminase reaction. So we converted the amino acid to alpha-keto acid to use in energy, and in that process we convert glutamate back to alpha-ketoglutarate. Pyruvate is converted to alanine. Alanine is then shipped to the liver, and then we run this reaction backwards. So this is your alpha-keto acid, and this is your, this is your alpha-keto acid. And alanine is your amino acid. So we run it backwards to make pyruvate, and then pyruvate can actually be converted to glycolysis, and then that is then sent to the liver to use to continue the cycle or to make pyruvate for more energy. So we send glucose, and then the muscle can use glucose to make pyruvate for energy, or we make more pyruvate to be able to allow us to break down more amino acids so we can continue running the alanine cycle. So once we make this pyruvate, that's where this pyruvate ends up. We make glutamate. Then this glutamate can then run through glutamate dehydrogenase. So remember, glutamate dehydrogenase, that's the one in the urea cycle. We convert the alpha-ketoglutarate back, and then we make ammonia, and then you have carbamyl synthesis, and then we go through the urea cycle, and then eventually we make urea to be excreted. So the whole reason we did all of this is we can use amino acids in the liver. Okay? So use amino acids in the liver, and they kind of just pass this amino group. We pass it to glutamate, glutamate passes it to alanine, alanine goes into the liver, alanine passes the amino group back to glutamate, and then glutamate sends the amino group in the mitochondria, and then you have the carbamyl phosphate synthetase, and then you have urea cycle, which then eventually excretes the amino group. Okay. So I know it's a lot, but this allows us to break down amino acids in the muscles. So alanine cycle makes glucose, but also transports nitrogen to liver. So that's the whole point of the alanine cycle. You form pyruvate in the liver, and then pyruvate in the liver can also be used in TCA, or we can do gluconeogenesis to make glucose back, and then send that glucose back to the muscle. Okay. So mechanisms of eliminating nitrogen. So this is alanine, so this is amino acid. This is alpha-ketoglutarate. This is a transaminase reaction. So alanine is converted to pyruvate, and in that process, it gives this nitrogen group, the alpha-ketoglutarate, which then becomes glutamate. And then glutamate can then donate the amino group to oxaloacetate to form aspartate, which then can enter into TCA. So what it's talking about is another transaminase reaction. Aspartate. We make alpha-ketoglutarate dehydrogenase. So we make, so this happens in the liver. Once we take glutamate, we take oxaloacetate. We convert it to aspartate, so this can enter the mitochondria and then be used in TCA using the malate aspartate shuttle. And in that process, we'll be able to get rid of our nitrogen group. Okay? So we can run this direction, and then we can also take glutamate oxaloacetate, make aspartate, and aspartate can then go into mitochondria for TCA. Okay. Let's talk about glutamine. So glutamine has 2 nitrogens. Glutamate has 1 hydrogen. They're both amino acids. You can convert between glutamate to glutamine using 2 different enzymes. So you have glutamine synthetase, so that the glutamate converts into glutamine. And then when we want to use glutamine to make energy for the cell, you run through a reaction called an enzyme called glutaminase this glutaminase releases this nitrogen group, which then can then go and be used by carbamyl phosphate synthetase, so CPS, and then be used in the urea cycle. And in that process, you make glutamate, and then glutamate can then be converted back to alpha ketoglutarate through glutamate dehydrogenase, then make an ammonia, and then this ammonia can then be also used by CPS for the urea cycle. Okay? So that's how you get rid of the two nitrogens in glutamine for to be excreted in the urea cycle. So these two reactions run opposite directions. They have different locations in the liver, and this actually prevents futile cycles. So some cells will express the glutamine synthetase, and some cells will express the glutaminase. In different cells, they're both in the liver, and this prevents a futile cycle. So if you're ever asked about a question about different cells expressing different enzymes in the same tissue, this would be a really good example, and it allows the cell to prevent a futile cycle. Okay, let's talk about the other reactions. So glutamate can then be converted to alpha ketoglutarate. We can also run glutamate to alpha ketoglutarate. Instead of using glutamate dehydrogenase, we can use a transaminase. And in that process, our alpha keto acid is oxaloacetate, which is converted to aspartate. Remember, aspartate gives the second urea, second ammonia, second nitrogen group. I don't know why that was so hard to say. So it gives us the second nitrogen group in urea, which then can be run through the urea cycle to eventually be converted to urea and then excreted. Okay, and then alpha ketoglutarate can then be converted back to glutamate. So this is transaminase reaction. Okay, so that's the whole reason I included this slide is just to show you about how different locations of enzymes, different locations in the tissue of different cells can express different enzymes to prevent a futile cycle. Mainly talking about glutaminase and glutamine synthetase. And it also helps get rid of the two nitrogen group for glutamine. Okay. So let's talk about some regulation. There's not much regulation. So we already talked about ADP and ATP affecting glutamate dehydrogenase. So when glutamine is converted to glutamate, remember we make urea. Urea can then go back and activate this glutaminase. Okay. And then glutamate can then be converted into a substrate called N-acetylglutamate, which then acts on CPS, our rate-limiting enzyme, and it can activate it. So remember, CPS takes ammonia, takes bicarbonate. So remember, we take bicarbonate, and it also takes ATP, and then we make carbamyl phosphate using all of that. So from glutamate to N-acetylglutamate, we use an enzyme called N-acetylglutamate synthase. This enzyme uses acetyl-CoA, and it's also activated by arginine. And then N-acetylglutamate can then allosterically activate carbamyl phosphate synthetase, which is right here. And then when glutamate is converted to glutamate, NH3 is lost, which then can activate the glutaminase enzyme. So this is referring to this up here. Okay, let's talk about our transporters in this lecture. So we have a transporter called SLC. They're a family of transporters. So remember, we have CPS, we take bicarbonate, we take ammonia, we use ATP, and then we make carbamyl phosphate. Carbamyl phosphate comes in with ornithine, makes citrulline. Citrulline leaves through the SLC transporter. Citrulline is sent to the cytosol, ornithine is sent inside the mitochondria, and in that process, we pump a proton out of the cytosol. And then ornithine continues through the urea cycle, and then eventually urea is excreted, ornithine is generated, and then it's sent back into the mitochondria to continue the cycle. Okay, and then, like I said, in that process, an H plus is pumped into the cytosol. Fumarate can be converted to malate using fumarase. So we already talked about this one, and then malate can then be converted into oxaloacetate using malate dehydrogenase, and then these are cytosolic versions of the enzymes in TCA. Okay, let's talk about nitric oxide. So this is the last thing I think I have on here. So if you've never looked or heard of it before, but nitric oxide is a strong signaling molecule. You'll talk about it if you take cardio-phys or if you take neurobio. Talk about nitric oxide a bunch. It can cause blood vessel dilation. It can also cause smooth muscle relaxation by making cyclic GMP, and it does this by interacting with guanylate cyclase. So this is really similar to adenylate cyclase, except it's guanylate cyclase. So instead of making cyclic AMP, we make cyclic GMP. Macrophages also use nitric oxide, so they use it as a superoxide to help kill bacteria. It can also act as a neurotransmitter. And the enzyme that does this is nitric oxide synthase. So if we go back, remember we said arginine can go through arginase, converted to ornithine and urea. So this same arginine can be converted into nitric oxide. So arginine has different fates, so it can be converted into nitric oxide using nitric oxide synthase, and then it can be converted to urea using arginase. So nitric oxide synthase uses NADPH. Using oxygen, we make nitric oxide. We also make citrulline. So citrulline can continue urea cycle. And then the same arginine can also run through arginase, and then it can be converted to citrulline. And instead of nitric oxide, we have urea instead. Okay. So nitric oxide synthase. So it's a homodimer. But the monomers are inactive. Because they have limited ability to transfer electrons. The homodimer is active, so homo just means same, dimer just means two subunits. Those two subunits are activated when they come together. So when monomer 1 and monomer 2 come together, they become activated. This is similar. Remember we talked about fatty acid synthase, similar to fatty acid synthase. The second enzyme in fatty acid synthesis. It uses cofactors FAD, FMN, and BH4, and the mechanism for it is we transfer electrons from a reductase to heme to oxygenase. So it has different isoforms. So we have eNOS. So the NOS stands for nitric oxide synthase. So we have eNOS, nNOS, and iNOS. They both make nitric oxide synthase. All of them make nitric oxide, but they both are expressed in different, depending on the tissue or the cells they're expressed in, and what the nitric oxide acts on. Remember we said nitric oxide is a signaling molecule. So depending on what the nitric oxide acts on, they have different names. So for example, iNOS will mainly make nitric oxide to act in inflammation. That's why it's called iNOS. So it helps stuff in inflammation. And then eNOS and nNOS have different functions, of course. Another difference between them is eNOS and nNOS. They have calcium is able to increase the affinity for calmodulin, which is a protein that binds calcium in the cell, and it allows it to act as a molecular switch to allow electrons to flow. So you can see that calcium is able to act on these two and increase affinity for it to basically make nitric oxide. iNOS is different. Calmodulin is a protein that binds calcium. So for eNOS and nNOS, when calcium is able to bind the calmodulin, this is able to increase the affinity of eNOS and nNOS. In iNOS, calmodulin binds at very low concentration, so it doesn't have much of an effect. Little effect. Okay, so it has little effect. It has little effect on the nitric oxide synthase. So this is another difference between the eNOS, NNOS, and iNOS. Okay, let's talk about the nitric oxide reaction. We're almost done here. So we have arginine. Remember, arginine has a guanidino group. The first thing we do is we hydroxylate this guanidino group on arginine, so we get this OH, and then we get this intermediate. We can then oxidize the intermediate to citrulline, and in that process, this hydroxylation that we did on the guanidino group is then converted to a nitric oxide when it's clear. So we won't go through the mechanism of this, but it uses a lot of electron transfers, and then just know that this can produce reactive oxygen species. So if this is ever asked about how is this related to like the pentose phosphate pathway, remember the pentose phosphate pathway can make NADPH, which helps make glutathione, and glutathione helps with reactive oxygen species. So if you're asked about nitric oxide synthase and how that's related to the pentose phosphate pathway, you'll have to talk about the oxidative state of oxidative phase of the pentose phosphate pathway. So that's one way that this reaction is related to other pathways that we'll talked about. Okay, so this is the mechanism for it. It's really complicated. I will not go through all of it, and also I don't know it that well, but I have it here if you're asked about it. And the reason I don't want to go over it is because I don't want to say something wrong, because I don't remember much about this last year, so I don't want to, and I don't have notes on it, so I don't want to give you something wrong. But this is the mechanism for it. Basically, you have two successive monooxygenation reactions. You have an iron-containing heme that helps coordinate with oxygen, and the important thing to know is, like I said, it has a lot of electron transfers, and this can produce reactive oxygen species. Okay, so let's talk about some stuff that nitric oxide can do. It can be acted upon by calcium and calmodulin, and those can help activate nitric oxide synthase to make nitric oxide, or nitric oxide can also diffuse through the membranes. One thing that it can act on is guanylate cyclase. So guanylate cyclase is very similar to adenylate cyclase, except guanylate cyclase makes cyclic GMP. Cyclic GMP can then act on PKG, so this is different. Remember, we had cyclic AMP, and it can act on PKA, right? Yep, it was PKA. So cyclic GMP act on PKG, and then PKG can have different effects depending on where it is and what tissue it's in. So for example, in smooth muscles, it causes relaxation. It can also act on transcription factors, change gene transcription of that. If we're in platelets, if we're in the blood, we'll inhibit platelet formation. And like I said, so there's different nitric oxide synthase, but iNOS affects inflammatory signals, and it does that by acting on NFK beta, which I think you'll talk about in cell phase if you're taking that class. Okay, let's talk about some disorders of urea cycle. Basically, you can have disorders on any of the enzymes used in urea cycle. So all of them except for one. All of them are autosomal recessive except for ornithine transcarbamylase, so OTC, and that one is X-linked. So all of them are autosomal recessive, and deficiency can occur in any of these. So we can get it in the N-acetylglutamate synthase, so that's the one that makes us N-acetylglutamate. We can get it in CPS, our rate-limiting enzyme. We can get it in OTC, ornithine transcarbamylase. We can get it in argininosuccinate synthase. We can get it in argininosuccinate lyase, or we can also get it in arginase. Okay. So whenever you have any of these disorders, usually you remember the whole point of the urea cycle is to be able to use amino acids for energy and then get rid of the nitrogen. So if we can't use the urea cycle to get rid of the nitrogen, well, the body won't be able to store as much protein as it can. So, and then this also blocks elimination of nitrogen. So ammonia then, if you eat a lot of protein, ammonia will then start to accumulate because proteins have amino acids and amino acids have the nitro group. So this ammonia accumulates without it being excreted as urea, and it can cause a lot of things in the nervous system. For example, it can cause seizures, and it can cause hyperammonia in the blood. So the therapy for it is you reduce your protein intake to reduce the amount of amino acids you have to transaminase, and then you can also supplement beyond the blood. What that means is if you have a deficiency here, you can supplement substrates here to continue the cycle without having to worry too much about it. Okay, so those are two different therapies for it. Okay, so this is the summary. So urea cycle occurs in liver. Some organs have not suppression, expression of some of the enzymes. Remember, this it helps regulate nitrogen elimination. And then cytosolic substrates are used to make nitric oxide. So this is talking about arginine running through the nitric oxide synthase reaction to make nitric oxide expression. Okay, so this is all for this. I'll upload it. I'll also upload the summaries for it. And if you have any questions, you can email me. I'm gonna try to reach out to Dr. Kanapka and ask her about review sessions for you guys and figure out dates and stuff like that. Okay, thanks so much for watching, and good luck with all classes. Good luck on the test, and have a good day.